and greetings from Potchefstroom, South Africa. Thank you all for attending Emma's Fryer's Master's Thesis presentation. I know it's early, so I appreciate you guys attending via Zoom and in person. I'm Nishi Rajakaruna, and I have had the great pleasure of being Emma's thesis advisor over the last 2.5 years. Professors Dina Grossenbacher and Tim Bean from the Biological Sciences, as well as BLM botanist Ryan O'Dell, serve on Emma's committee. Emma received a BSc in botany from Humboldt State University and then worked as a field botanist and environmental planner for 2 years prior to starting her MSc. Emma's research, as you will hear today, focuses on the factors and mechanisms driving the assembly of a very special yet understudied plant community found on salt and clay-rich soils in Central California. Emma, in my opinion, had a really well-rounded graduate student experience. She raised her own research funds by receiving 13 regional and national grants and scholarships. That's pretty impressive. Served as a teaching associate for 5 biology classes, including general botany and plant taxonomy, and supervised research of 6 undergraduate students, including 4 Frost summer research students. Emma is obviously very passionate about vascular plants, but she's also into bryology and lichenology, and always takes great pleasure in sharing her love for natural history through teaching, mentoring, and her amazing botanical illustrations. Emma has presented her graduate research at regional, national, and international conferences and was recently accepted to 2 fully funded PhD programs at Stanford and ETH in Zurich, Switzerland. She is soon going to start a PhD in the plant ecology group at ETH. So I would now like to invite Emma to tell you all about her findings on the factors driving the striking patterns of plant diversity at Carrizo Plain National Monument and adjacent areas. Emma, take it from here.
All right, thank you, Nishi, for actually kind of making me nervous now. So first up, is everyone on Zoom able to see Nishi? You guys have my screen still? All right, so
yeah.
Great. So I'm really excited to talk to you all about this really, really cool system that I've been working with. My thesis project has been focused on this community of native annual wildflowers found on the Vertic clay soils of the San Joaquin Desert. And I'll explain what I mean by Vertic clay soils in a minute here. Slide progression. So my study species are found here in the San Joaquin Desert. And so that includes the southern portion of the Central Valley and then also the eastern edge of the Coast Ranges. And this project has been kind of big. I've mainly been looking at how edaphic niche, so the soil niche of these species in competition, are really shaping this plant community. And while our focus with this community is largely related to these unusual soils that they're found on, I do kind of want to give you some background and context for this study system first. So this community was initially interesting to me for several reasons, but firstly, it was interesting because this is a part of California's flora that's been kind of overlooked. I grew up in Fresno here on the eastern edge of the San Joaquin Desert, and I didn't know about this flora. It wasn't on my radar well into adulthood. And so when I first became aware of this community while I was working in the region as a field botanist, I was also kind of surprised to find out how abundant wildflower blooms formerly were in spring in this area. And so native annual forbs formerly made up a much more substantial proportion of the region's flora before it was really dramatically transformed by agriculture, oil extraction, and invasive annual grasses. And so the first description of this region before this transformation kind of took place that I ever read was actually by John Muir, who called it like a lake of pure sunshine, 40 or 50 miles wide, 500 miles long, one rich furred garden of yellow composite. And then in a letter that year, again, he called it the floweriest piece of world I ever walked, one vast, level, even flower bed, a sheet of flowers. And then 20 years prior, a man named Thomas Jefferson Mayfield passed through the region with his family as a child. And later in life, he described it as like a crazy quilt of color where the quilt was flowers, rows and rows of rose, yellow, scarlet, orange, and blue, each species attached to a different soil type with some colored bands stretching a mile across. And this is probably not what you pictured when I showed you that map of the San Joaquin Desert. Today, we see far fewer blooms with less intensity than in relatively recent history. And so this photo was taken outside of Bakersfield in Kern County sometime in 1942. And then this one was taken in 1936 in the same general area, somewhere outside of Bakersfield. And this photo up here is actually the oldest photo that I'm aware of that shows the region before it was really developed. And it's kind of hard to tell, but these are not annual grasses, right? They're not tall enough or light enough in color. And the description of this photo is something to the effect of gathering flowers. And so it's kind of interesting, having grown up in the Valley, to learn all of this. It was a very like dramatic realization to have, having only ever like traveling through the Valley, having only ever really seen these vast swaths of agricultural land, and all of these locations today are really dominated by invasive annual grasses. This is probably what we all picture when we think of that area, to the point that this region was previously described as grassland. And it was only in the paper that first sort of outlined the San Joaquin Desert that I first saw how severe the degradation of the region was. Something like 60% of the San Joaquin Desert has been kind of like permanently transformed by human activity. And then this region has also had not just humans really changing the landscape, but also invasive annual grasses like Bromus madridensis. And this is one of the main invasive species in the region, and it's been really impactful across the Western United States. And so it's while this sort of remnant nature of this plant community that initially kind of drew me to this project, this community is also interesting for a couple of other reasons. So firstly, these species are growing on vertic clay soils, which are a really stressful substrate for plants. And as we know from the ecology of serpentine species, stressful substrate can often lead to endemism. And that's a really important component of California's plant diversity. It makes up kind of a disproportionate chunk of the plants in California. And while serpentine plant communities have been pretty well studied, there's been a lot of work on them relating to their evolution, speciation, and community ecology, and they've been a really valuable model system for plant ecologists. Vertic clay soils, however, have been pretty much unstudied in this regard, despite the fact that they're similarly stressful substrate. And we really don't know a lot about their ecology. So what do we know about vertic clay soils? So these soils are uncommon, and vertic refers to the properties of the soil. These soils are found more in California and Texas than anywhere in the United States. And we see them in California along the coast and the Coast Range, and in the San Joaquin Desert. We actually have them quite often here in San Luis Obispo as well. And these soils are, and these soils have really high clay content, specifically smectite clay, and a minimum of 20%. And that's a distinction that's important because smectite is a specific mineral type of clay. And that they have much higher capacity to absorb water than other types of clay like kaolinite. And so because of this, because they can take up so much more water, these soils expand a lot when they're wet and then shrink down a lot when they're dry, which is what creates this really characteristic cracking that you're seeing here. And so this is a potential source of physical stress for plants, because if a plant is growing in this soil and that soil begins to contract, you're potentially getting plant roots getting torn. So this is a source of physical stress. And then high clay content of any kind is also going to potentially introduce water stress. So clay particles are extremely small. This is a smectite clay particle. This is about 1 micron in diameter. And because they're so small, in contrast to sand and silt, which are many orders of magnitude larger, for any given volume of clay particles, you're going to have a lot more surface area per that volume than you would if you had larger particles. And the space between those particles is also going to be smaller. And so this means that the water tension of the water that's between those clay particles is going to be a lot stronger as well. And this can ultimately mean that these high clay content soils can have much lower water potential than plant roots and can actually keep the plants from taking up that water, even though it's right there. And then these soils are also frequently high in sodium. So in the San Joaquin Desert, where we see vertic clay soils, they're often in areas where the geologic formations are marine in origin. And so we've got these clay soils weathering and inheriting high sodium from the parent material, which is typically marine shale. And then if all that clay runs off into basins, those dissolved salts can accumulate, like we've seen at Soda Lake, if you've ever been out there. And so overall, these are really stressful, extreme soils. They have a lot of potential sources of stress for plant life. And so while this is this really challenging substrate that suggests that there's some really interesting ecology going on, basically no work has really been done on how plants interact with these soils outside of like agricultural applications. And Over time, however, botanists have observed the apparent association of some species with these soils and the restriction of some of those species to these soils exclusively. So we have this element of edaphic affinity in this community as well. And so my project is focused on 12 species that have this affinity for these soils. And of these, 7 are considered rare by the metrics that the California Native Plant Society uses. So I'm not going to go into their ranking system, but basically 1B1 and 1B2 CRPR's California Rare Plant Rank. These 2 rankings are kind of the rarest you can be without being extinct. And then this one is undescribed, but probably meets that same standard. And this is the only one that's a bit less rare. So while you can look in the Jepson e-Flora in their ecology notes, and you can see clay mentioned for many of these species, and the California Native Plant Society's Rare Plant Inventory also often references when clay is associated with the species. We don't really see a lot of information otherwise. Some species will have specific epithets like Calochortus argilosus, which we have here in San Luis Obispo. Argil means clay. And so this has been observed before, but no one's ever really quantified it. And so I've started a little side project with a couple of undergraduates, Hannah Huntley and Alyssa Shawn primarily. And we've been going through about 200 species that have suspected clay affinity. And so far, actually looks like California does have a lot of species that have these really strong association with clay soils. So what we've been doing is we've been pulling all of the herbarium records from California for these species. And so for Benetoa occidentalis, that was 150 records total. And we've narrowed it down to just the records where you could actually tell what substrate those species were growing on. In this case, that was 63 records where they stated the substrate. And then of those collections, we found what percentage of them were actually referencing clay soils. So for this species, 57, which meant 90.5% of cases where we knew the substrate, they were growing on clay. And so it's a pretty strong case for probably endemism to clay soils. And we did this for all of my study species. And so that's what those percentages here are telling you about these species. And so we found that these records reflect pretty strong affinity or association to vertic clay soils for these species. And particularly given that botanists, most of them aren't very good at soils, and a lot of them don't record substrate in their herbarium collections. But it's also true that clay is probably getting noted less often than more charismatic and readily identifiable substrates like serpentine. And so while this did not make it into my thesis proper, I've included it here because I think this is important context for this community and because it supports our idea that these species had vertic clay affinity. Now, these species were also selected because they just occurred at my field sites. So both my field sites have vertic clay soils. I have one at Cantua Creek here, and then another at Carrizo Plain here. And both these sites also have this really distinctive floral mosaic pattern that we're seeing here. We've got these distinctive kind of single species patches. And Ryan Odell, who's on my committee, is a BLM botanist who's worked the area for a long time. And I've had the benefit of his experience with this system and his experience. And it did look likely that these patches that we're seeing here, which are pretty discrete, actually reflected differences in the soils. And so we thought that these soils were about as heterogeneous as that pattern of bloom suggested. And if we look back, that observation had already been made before. Each species attached to a different soil type. And these species patches that we're seeing that are really dominated by a single species can be much more visually dramatic following very wet winters like this year. And this has certainly drawn some attention in recent years. Super bloom mania seems to have reached a sort of fever pitch. And so we'll probably be seeing a lot of people in Carrizo very soon. So my study system has this combination of factors that makes it really interesting. We've got these species that have this strong affinity for this really stressful substrate. We've got this very mosaic patchwork-like pattern of these blooms that we think reflects heterogeneity of the soils. And then all of this is going on with this invasive annual grass in the region that's really transformed the area. And this is a particularly interesting combination of factors, in part because of the heterogeneity. Heterogeneous landscapes, like at my study sites, are kind of by default going to entail a higher number of possible ecological niches. So by niche, I just mean the biotic and abiotic conditions under which a species can survive and persist. So a species is going to have some specific range of environmental factors under which it survives. So if if we have this landscape where we have all these soils, these colors, and if this species, Monolopia major, can only survive, say, on all of these soils that are here in the foreground, then, so just these soils, then that's going to be the species' fundamental niche. So the conditions under which it is able to survive and persist. But in any actual habitat, we're going to have biotic factors like competition, which are going to affect the species' ability to survive on some of those soils. And so that's going to narrow this fundamental niche down to a realized niche. So even though it could survive and persist on all of those other soils without competition, when you account for biotic factors, these are the soils we actually see it surviving and persisting on. And that realized niche is also going to be the one where we actually see these species in their communities. And so that's basically what we thought was going on with these mosaic bloom patterns that we're seeing, that these are a reflection of these species' realized edaphic niches. And so if that's what's going on, then there are some really cool ecological questions that we can ask about this system. But since there hadn't really been any work done with it before, and I kind of wanted to like establish that this was actually what was going on. No one had really looked at it before. So do these bloom patterns actually reflect distinct realized edaphic niches? And so that's basically the first part of my project, is just confirming what's going on in this system. So if those patches do reflect edaphic niche, then we would expect to see differences between the soils between those species patches. And so I split my project into a field component, confirming that this community was everything we thought it was. And then I had a greenhouse study, which was more accurately called a pot study, because it was just in a backyard with no greenhouse, to answer additional questions once we established those facts. So for that field work component, I went out to my sites and I found patches where we had a single species clearly dominant and collected soil samples from the centroid of each of those patches. And I should be clear that like most of this was Ryan at one of my sites doing the really hard work. And then at Carrizo Plain, it was a lot of really hardworking undergrads who were troopers when we had to do some of this in summer. So it was not just me. But for each species, I did this for multiple distinct and separate patches. And I did that for a minimum of at least 5 patches per species. So once I'd done this for all 12 study species, I had all of these soil samples tested for properties that were physiologically relevant to plants, and then for some of the properties that we thought were likely to be acting as stressors. So pH, and then nitrogen, phosphorus, potassium, calcium, and magnesium are like basic plant nutrients. Sodium's kind of an obvious plant stressor to look at here. CEC refers to cation exchange capacity, which is just the ability of a soil to hold positively charged ions, which affects plant and mineral uptake. Calcium to magnesium ratio is important in that they can compete for uptake by plants. Excuse me. And then sodium to potassium ratio, sodium's a stressor, but also sodium can potentially block potassium uptake depending on that ratio. Clay percentage was important for maybe obvious reasons, but also because it captures that potential source of water stress. And then coal here refers to coefficient of linear extension. And so that's basically just a measure of shrink swell potential. So that shrinking on drying that caused that cracking. And so what we did was we basically saturated one of the soil samples, extruded it through a wide syringe and measured the length. And then we let it dry, measured the length again. And that difference between the two gave us a measure of that shrink swell potential of the soil. So that physical stressor, and Alyssa, Sean, and Skylar McKinnon did a lot of this. So I just want to say thank you to them. So since all of these properties can affect the soil, and soils are kind of complicated things to describe and understand, I really wanted to capture as much information as possible about them. And so I did a principal component analysis with all of these soil samples data with all the information about these properties. And what this is doing is basically just distilling the majority of the variation that's explained by these properties down to fewer axes rather than a single axis for every single one of these, which would be awful. So in this case, we have 34% of our variation explained by this axis and 25% by this axis. So just with these two axes, we can look at over 50% of the variation in this data. And so this first most explanatory principal component here was most defined by phosphorus, sodium, cation exchange capacity, and that sodium to potassium ratio. And so as we move this way across the plot, we've got soil samples, these points getting higher in those properties as they go this way. And that second principal component here was defined most by calcium and magnesium and the calcium to magnesium ratio, with calcium increasing and magnesium decreasing as we move down this axis. And so this entire area here that we're looking at with all of these soil samples plotted in is just the space of all possible combinations of those soil properties and where those soil samples fell within that space. Now, if you saw my map earlier and you've been wondering how these two study sites actually compared, given they were kind of physically disjunct, these are those points with 95% confidence ellipses for them by site, so which site they were collected from. And so this is Carrizo Plain, and then this one's Kintua Creek. And we can see that they overlap a lot on this axis, so they're fairly similar in terms of calcium and magnesium and the ratio of the two. And on this axis, they're a little more removed from one another, so a little more different in terms of these properties, but we still do see some overlap. And then if we look at all of these points distinguished by species, so which species patch the soil sample was collected from, it's not great to interpret. I tried a lot of different ways, but 12 species is just a lot to have on one plot. And so we're instead going to look at it just one species at a time for a few examples. So if we focus on Lepidium druidii, subspecies druidii, this one extended really far out this way, away from the other species, and it spans this really large area across this axis. So it spans a wide range of these properties. And in this case, it was actually extraordinarily high sodium content that was really pulling it this way away from those other species. And in some cases, that was like 15,000 parts per million or more, which is incidentally about how many parts per million sodium the human body is. If we look at our other subspecies of Lepidium druidii, which was subspecies album, it has a much narrower range in terms of this axis than that last one. It has a much broader range on this one, so a broader range of calcium and magnesium in that ratio of the two. Some species had a much narrower range of soils, so this is Leptosne Calliopsidea, and it covers a lot less soil space here. So this is a much narrower range of soils that it occupies. And while some of these species overlapped in soil space, others didn't overlap at all. And so we're seeing the apparent realized edaphic niche for these species and how they compare to one another. But 12 species, again, is a lot to try and look at once. It's difficult to eyeball this plot. And so how can we test for actual differences in these species' realized edaphic niches? And so what I did was I took where each species points fell on one of these axes. So where the species points fell in terms of the coordinates on this axis, so that I could compare species based on those coordinates. And so this is all of those species soils plotted by where they fell on just that first principal component. And I did see a difference in their means along this axis. So we had a difference in niche, at least as it was defined by the properties that define this axis. But also, I found a difference in variance, so in the spread of these points. And so it looked like we also had a difference in the niche breadth of these species. And if we look at where the points fell on that second principal component, I had the same results. We saw differences in both niche and niche breadth. So it does look like these soils are different between species patches, like they reflect differences in the soil. Like these species patches reflect differences in the soil, excuse me. And we saw that these species had different realized edaphic niches and they had different niche breadth. So this community looks like it has this really cool situation where we've got edaphic heterogeneity combined with all of these other factors. And so part of why this heterogeneity is so interesting to me is because, speaking generally, an environment that has lower heterogeneity, so say we only had 2 soils, is going to have fewer niches available for species. And an environment that has higher heterogeneity is going to tend to have more niches available. And typically that's also going to mean greater diversity. But this also means that communities with high heterogeneity are going to tend to respond differently to things like invasion, say by Burma's madritensis. And so this sort of takes us to the second half of my project. So having established all of this about this system, I wanted to know what the fundamental niche of these species looked like. So what kinds of soils these species are surviving on when competition isn't present. And I also wanted to see, was Bromus madridensis reducing species performance? In this case, I did that with biomass, especially given, you know, it's been this dramatic change in the region, having Bromus madridensis move in. And still we're seeing these really like strong, almost single species stands of these blooms. And then lastly, I wanted to see, how does competition affect that edaphic niche optimum if it's there? And so given the results of my field work, this last point was especially interesting to me because these species are in stressful soils. In contemporary niche theory, the idea is that two species that are competing with one another, so we'll say these two colors are two species, and if they're under some environmental stressor, so we'll say this gray soil is really stressful, then that outcome is predicted to depend on those species' tolerance of that stressor. So if this lighter colored species has a greater tolerance for the stress than the other one does, we would expect to see it eventually completely outcompete the one that's a poorer tolerator of these soils. So I want to know if that could be what's happening here. Is that contributing to these pretty much single species patches that we're seeing? So to answer these questions, I did my pot study, and I did this with the same species that I'd sampled those soil samples from in the field to see the effects of soil and competition on these species in an experimental manner rather than observational. So for my treatment soils, I really wanted to capture the gradient of edaphic stress that was really present at these sites. And if we look back at that PCA and what really defined these soils, what really captured the differences between them, it was mostly these properties. So for the pot study, I used 3 soils that were collected for me by Ryan. And I just want to note it was like 5 tons of soil. So it was not like a small favor, him getting me them. It was a lot. So, and I have these soils referred to here by sodicity, and I have them referred to that way for the remainder of this presentation. And they did capture this gradient of sodium content, but they actually also captured a gradient of that sodium to potassium ratio, of the calcium to magnesium ratio, and of that cation exchange capacity. So they did capture a gradient of all of these things that really define that PCA of our field collected soils. So I planted each species across each of those soils. And then I repeated this both with and without a competition treatment. And so that treatment consisted of adding 3 Bromus madridensis plants that I seeded in the same pot as a study species, spaced evenly outside on like the edge of the pot. And then I did this with 4 replicates of each treatment combination, and then pots were randomized into 4 blocks. So we had 1 replicate per block. And I did this for all 12 species from seed to seed set from December of 2020 to July of 2021. So this is what I was doing like during peak COVID, basically the whole time. And I just want to say that my friend Luffy Bailey let me use his backyard to do this and basically like single-handedly saved this project. That was like when COVID first happened and like everything was chaos and no one knew if they had access to resources. And he just kind of let me like move in for 10 months. And I was there like all the time. So I just want to say thank you to Luffy for that. And this was a really big undertaking. And it turned out part of why it was such a huge amount of work was just because of this really extreme nature of these soils. So these soils absorb so much water that you can't just like put the soil in a pot and then water it from the top, because the water doesn't actually just percolate down. The first inch or 2 of that soil absorbs the water, swells, and then holds the water so tightly that you just get this like blob of saturated clay, and then like nothing under it gets wetted, at least not within a week, because I tried waiting and it didn't work. So I actually had to like hand mix water into these soils in bins and then put the wetted soil in the pots for 288 pots. It took a long time. And so after I got all of these pots seeded with my study species and my competition treatments, a lot of my time was actually just spent weeding out all the volunteer plants that were in the soil seed bank. And that was all done just identifying them by their cotyledons, which if you've ever had to do, you know, is tricky. It's very difficult. And then also these clay soils were so sticky that I had to weed them using watchmaker forceps. I couldn't use my hands because clay would just come away with my hand. And honestly, I'm just making you guys watch this because I spent like 4 weekends doing just this for 288 pots. So, but it actually went pretty well, right? So I actually had great success with germination. I lost hardly any replicates. And I got my own little bloom. It was really actually a really amazing experience to get to have that all in front of me. But once plants started flowering, I did not learn anything from how much work it was to set up this project. And I started counting how many flowers were open on each plant every day, which I did come to regret eventually, but this did not make it into my thesis, but I want to mention this data and also that we collected data on reproductive biomass, so reproductive output, specifically because Alyssa, Sean, and Skylar McKinnon here both did a Frost undergraduate research fellowship project with me looking at this. And so they were doing all of this really painstaking, tedious work collecting that reproductive output all summer. And they did an amazing job and they worked so hard and I'm very appreciative. So I just wanted to make sure you all saw how much they did because like, look at that. Brutal. That was their whole summer. So once flowers started to mature, they started dropping parts. And so I actually had to like capture all of the parts that they were trying to drop and all the fruits they were trying to drop in these adorable little wedding favor bags, just to make sure I didn't lose that biomass. And so as we started getting into summer, I tapered off the watering to reflect field conditions. And then as each plant senesced naturally, I actually separated them into the above ground and below ground parts. And I did this to get the biomass, so just dried the above ground parts, but for the below ground parts, the problem is they're still in all that soil, that very sticky clay soil. And so I actually rinsed the soil off the roots, which was kind of awful. It was horrible. It took months. I spent like 2.5 months doing this. And I did this partly because I wanted to look at root investment by these plants based on the soil stress. So it basically to see in more stressful soils, are these plants allocating more biomass to the roots? So is there a higher root to shoot ratio of biomass on stressful soils? And I didn't really see a strong pattern at all. So we're not talking about that. We're talking about total biomass today. And so with all of that biomass data, and these are not just random bags, this is all the plant material I showed up to San Luis Obispo with. There's more than that, but I first approached this biomass data at the community level. So I wanted to see whether species, soil, and competition predicted biomass in the study, and did we see differences in biomass by species and treatments. And so I took biomass for all of those species together in one model, and I did a linear model in ANOVA. And then based on the results of that model, and because these species had distinct realized edaphic niches, I followed this with individual species models. So looking at species one at a time to see how soil and competition treatments affected them. And part of why I did this was just so I could use the most powerful ANOVA based on the interaction significance in those models. So because these two types of models were generally consistent with one another, I'm just going to focus on the species models. So our first research question, do species have fundamental edaphic niche optima? So starting off just with Benetoa occidentalis rather than flashing all 12 plots at you at once. Our community model showed that we had a difference in biomass by soil type. So in this species model, in this plot, sorry, these are the soils here, and then this is biomass. In this species model, biomass was significantly different on at least one of those soils relative to the others. So it did look like we had some kind of edaphic niche optimum across these soils in this species. And my treatment soils may not have caught like the true soil niche optimum for this species, but we see evidence that one exists based on my treatment soils. And that was true for all species. And don't stress like trying to interpret all of this at once. It's kind of unfair, but this is just so you can see a general pattern in the community where we had at least one soil that was higher than at least one other soil. And so all of these species had some kind of edaphic niche optimum within my treatment soils. So when competition is absent, it does look like we have that niche optimum there. And in the community model, we actually had an interaction of species and soil. So the biomass of a species depended on which soil it was growing on. Now, moving on to our next research question, does Bromus madridensis reduce species performance? And so again, don't try and interpret all of them, but overall, just looking at them, the light gray is when competition is present and dark gray is when it's absent. You do see a pretty strong pattern here where we have much lower biomass on all of these soils when competition is present versus when it's absent. And we saw this in all species except for one where it didn't quite meet our significance threshold, but we see a really strong pattern in this community. And so it does look like Bromus madridensis is affecting performance in this community overall. And then lastly, how did competition affect edaphic niche optimum? So we saw that competition affects these species. We have some kind of edaphic niche optimum. And looking again just at one species, Phacelia ciliata for simplicity, in this species and several other species models, we also had an interaction of the soil and competition treatment. And so that just means that competition's effect on biomass, so the difference between this and this, actually differed in size depending on which soil you were on. And so we saw a much higher difference here than on this soil. And in this case, Phacelia ciliata's apparent soil niche optimum when there was no competition present was highest on our somewhat sodic soil. But when competition was present, it was actually highest on our non-sodic soil. And so this shows that competition can actually shift species edaphic niche optima. And we saw that interaction of soil and competition in our community model as well, but we also saw it significant in over half of our species models. And so these species competitive abilities, it looks like they can differ within the range of soil chemistry found at my study sites. And it looks like competition can actually cause shifts in edaphic niche optima, at least for some of these species. And so when we look at this really striking patchwork of blooms, these species patches do appear to reflect differences in the soil. Species do have different realized edaphic niches, and we see different breadth of those niches. And these species do appear to have fundamental edaphic niche optima. Competition from Bromus madridensis reduced performance for most of these species, but more than that, it can actually cause shifts in species niche optima. So this last point was especially interesting to me, given everything we know about this system, because it means that when we've got all of this local scale heterogeneity, which means more available niches, if we have some species that has a fundamental niche optimum on this brown soil, so say this brown soil is kind of low stress, this gray soil is higher stress, this species does best when it's on this brown soil, but its fundamental niche includes this more stressful gray soil. It just happens to do better on this one. That means that if this species is faced with a competitor that can do better than it can on that brown soil, it might be able to persist despite being outcompeted on this preferred soil by surviving in a realized niche on that less optimal soil where it's simply a better competitor than that other species. And so all of these results suggest to me that the heterogeneity on a local scale that we're seeing in this system might also be contributing to these species persistence despite this really intense invasion by Bromus madridensis and other invasive annuals. And that potentially this is also contributing to these really dramatic mosaic patterns of blooms that we're seeing. And just to be clear, my results are consistent with this, but they don't prove it. You would need population level metrics and kind of a different study design to really prove that this is happening. And I had limited soils in my study, and this was just a master's project. I had a time limit on me, but I don't know how realistic it would be to do that kind of like gigantic study to prove this is happening, because these soils, when they're wet, you cannot, it's not possible to drive on them. You can't get there. It's completely impassable. I'm not even sure you could hike on them. And so I'm not sure if it will ever happen, but I really hope that someone picks up work with this community after me, because it's been really incredible getting to be one of the first people to ever look at this like awesome system. And I guess I still have my side projects to look forward to, but thank you all so much. I do want to say I got a ton of funding that made this possible, especially in such a weird time as that first year of school during COVID. And I'm very, very grateful, because it would not have happened otherwise. And I'm not going to spend too long on this because I'm going to get emotional, but a lot of people also made this happen. So I'd like to thank first like my advisor, Nishi, and then Ryan, of course. Oh my gosh, he did like so much labor to make this happen. And committee in general for a lot of advice and revising and help in R and just like talking me through things. It was really helpful. My fellow graduate students, especially Stephanie, Nora, Michael, and Annie. You guys are all what like made this the grad school experience I wanted. Many faculty and staff, definitely more than I fit on my list here. And I really want to thank Dylan Armitage for a ton of work and support and encouragement over the last 3 years. Ken Kelly for a lot of encouragement and support, and also Meg, including just for letting me complain. And again, Luffy Bailey, like I lived in his backyard for 10 months. I'm very grateful. My undergrads and volunteers also, it's been amazing getting to work with undergrads on these projects. And like, I've enjoyed it a lot. And they did so much work, especially Alyssa, Skylar, Hannah, Chloe, and Gilbert. So thank you all so much. Any questions?
Thank you, Emma, for a wonderful presentation. Let's open the floor for questions. We've got plenty of time. It's not even 9 yet.
In-person question, yes.
For your principal component analysis, you had species that had a wider range, and I'm wondering if you saw those species taking up more area or volume when you were out.
So the question was whether species with a wider range in the PCA were covering more soils in the field. I wanted to try and tie the soils these species were actually found on in the field to the results from my pot study. For this reason, I wanted to see like how the two connected, and I could not find, I tried for like months, I could not find a good way to do it with the study and soils and setup that I had. I suspect you would see relationships there, but I don't have a great way to do it with the data that I have, unfortunately. If I did it again, that would be one thing I'd be trying to do.
Yeah, or just if you like saw it in person.
There were definitely some like overall trends I noticed that were consistent. I would probably have to like look at it and sit down with it for a minute to really give you like specific examples, but the lepidium that went way out into the sodium was not a surprise based on what that site looked like.
Emma, there's Andy in the audience who has to leave in 5 minutes. So can I direct his question to you? Andy, go ahead.
Sure, let me jump off the mute on there. Yeah, can you hear me okay?
Yes.
All right. I was just kind of curious on the irrigation side. Were you using tap water or were you filtering it?
I was not, and it was well, whatever they put in the hose, I guess, in Fresno. So it was not. And I did overhead watering as well as they were in sub irrigation trays, which did mean that probably that bottom inch of soil was basically anoxic, but it kept them watered. And the volume of water that was going on, I don't know how realistic it would have been for me to filter it, because I did consider it. And I think I was kind of at my max capacity for that setup at that point.
Totally. I know like the nursery setting, we kind of struggle with that sometimes. Like the municipal water can kind of build up over time. So yeah, interesting. Thank you.
Jen?
Emma, were all of your soil samples on slopes? And if they, like this picture, right, everything has, it's on a hill. And do you think things are different if they were flat?
So I included the slope and aspect in one of my earlier PCAs. And I used like a northness, eastness thing for like incorporating aspect as well as slope. And they weren't actually contributing hardly anything to the PCA, which was surprising. Maybe I could have just kept those in there to put that in the results, but kind of had to graduate eventually. So.
And was everything on slopes?
Nope. Everything at Carrizo was flat. And I really thought it would be more important, but it wasn't. Yeah.
In your observations, have there over the last couple of years, or conversation that you've had with other botanists, do the boundaries of those patches change over time, or would you expect them to?
I grilled Ryan about that a bit when we were in the field, and he doesn't think so. And if you look at aerial footage of Carrizo over the last few blooms, they're pretty consistent there. Yeah, because I actually like dug really deep and like got a trial membership to this like satellite company to like check the imagery. And yeah, it actually looked really consistent at Carrizo. I could not get good imagery for Cantua Creek, but Ryan had photos over several years and they were pretty consistent.
Yeah, I was thinking about this year specifically with salt and soil, whether or not all of this rain might like kind of change the salinity of the soil and patches, how that might affect.
If I won the lottery, I would probably try to do a long-term study and like not bother with school again, but yes.
I don't think you recorded data on this, but sort of anecdotally, did the bromus have a preferred soil in your experiment? And also, did it have a preferred competitor? Like, did it do better in pots with any species?
I do suspect that the study species had different effects on it, and I really wish I had done pots that were just seeded with Bromus magitensis, so I could do that, because I could have gotten that biomass data easily, but I didn't, so I couldn't look at that. Yeah, I mean, easily in like the sense of my project's norms, but I did notice it seemed like it slightly preferred the sodic soil. We did see an overall pattern where most of those species did. And actually, if you look at the PCA, you do kind of see all of them centered at a similar point to where that sodic soil fell within the PCA.
Emma, can you repeat the audience questions to the Zoom audience?
Yes,
If you don't
sorry,
mind.
everyone.
All right, thank you.
Emma, it looked like most of your species preferred the medium soil.
Mhm.
And so you didn't find, species that like had a low sodic advantage and species that had a high sodic advantage. And so I'm wondering how you explain those results, given the patchiness you see in nature.
Do you mean that all of my you're saying all of my species looked like they did better on the sodic soil? It was actually not. So Jen just asked, it looked like most of those species did better on sodic versus the other two soils. And how do I explain that? But actually, we had a couple of species where that was the lowest biomass was on the sodic soil. And so I do think part of this came down to just like a limitation of my treatment soils, and that was part of why we saw that pattern. But I also think that if I had had more of a gradient of treatment soils, we probably wouldn't have seen that convergence quite so strongly. But that's definitely like an overall pattern I observed.
And did you put the chemistry for your pot soils on the same PCA?
Yes. Would everyone like to see that PCA?
Yes, please.
Okay, if my mouse can work. Oh my gosh. All right. Let's just go back a billion slides.
Your
Wow, my
graphics
mouse-
are beautiful, by the way.
Thank you. I worked really hard on them. We'll get there. My mouse decided to stop working. Sorry, everyone. Both my mouses. Wow, I have so many slides. I apologize. Okay, so let's go to a clean one. All right, so this point, and this point are my treatment soils. So not quite the ideal distribution of them through that PCA, but given the limitations on me, I'll take it.
Yeah, that's really great. So your pot experiment is really looking at the gradient of PC2 much more than it was PC1. That's very interesting.
It would be potential, sorry to jump in, it would be potentially interesting to look at the deviation of the field soil from your experimental soil and ask how much that explains competitive outcome. Sorry.
Yeah, that's actually something I've like poked around with. I did get the advice at one point that maybe it was time for me to move forward in this project though. Now that I'm done defending though, I can do whatever I want, so. Any questions left on Zoom?
Just want to say it's a fabulous study. You've done an amazing job. Congratulations.
Thank you. I see a question in the chat. So I'm curious about the presence or lack thereof of native grasses and any anecdotal observations you have in the context of your site. From memory, I don't recall native grasses being very significantly present at any of my sites. I did actually make notes on like what species were present and roughly what % cover were present. And really the only, I think the most common grass that I was seeing was the brome. But really it seemed like these soils are too extreme even for some of the more tolerant like perennial native grasses. It would be interesting to go back and kind of look and see if there's a difference in how native versus invasive grasses are responding, but I guess, again, limitations of the study.
All right, if there are no other questions on Zoom, it's 9 o'clock. Maybe we should conclude at least the Zoom session. And I know you guys have celebrations in person, and I'm sorry that I'm going to miss it, Emma. Yeah, thank you all for attending, and thanks, Emma, for a great presentation. And please join me in giving Emma one round of applause for doing a fantastic job. Thanks so much.